Thank you for being here. I hope everyone is safe and healthy. I want to introduce you a great news and ask you to save the date. Igenomix, it's organizing, Igenomix Foundation, it's organizing a symposium, a 4 hours symposium, where you'll be able to do the inscriptions for every presentation as a webinar, or the whole symposium at once. This will be on September online, and the first one will be transmitted from US, United States. But our idea is to have a small symposium like this with 4 hours every quarter. And we will transmit this symposium every quarter from a different place in the world. As you know, Igenomix have been working worldwide. So the idea is have the first transmission done by USA, the second one from Europe, the third one from South America, and the fourth one, when we will be completing 1 year of the symposium, be transmitted from Asia. So I hope you can save the date and keep in mind in the next webinars, we'll be coming for you with more details, and we'll be bringing the scientific program, and you'll be open to find, of course, in our webpage, all the information that you want and that you need. Please save the date, September 17th. So today, we have our second presentation on our webinar series, Reproductive Medicine, What is New? Last week, we had Dr. Carmen Rubio. Today, we have Dr. Antonio Capalbo. We will still have Dr. Felipe Vilela and Dr. Carlos Simon closing the webinar series. But for today, we have Antonio Capalbo. I know that most of you know him from his publications. He doesn't need any introduction, but following the instructions, I have to tell you that Antonio, it's PhD in human genetics. He's also an embryologist. Nowadays, he's scientific and laboratory director in Igenomix, Italy. Also, Antonio, it's a member from the special interest group on ESHRA in genetics. Antonio, it's our pleasure to have you here and list what you have to tell us about mosaicism. Thank you so much.
Thank you, Juliana, for the nice introduction. And thank you, everyone, for joining us in this webinar. And I know there are also a lot of friends and colleagues joining. I'm going to spend the next 40 minutes with you discussing about this hot topic, I think, in our field, in PGTA and IVF in general, which is about mosaicism. And during the first part of my presentation, I'm going just to see the main challenging that we have and we face when attempting to diagnose mosaicism from a single trophectoral biopsy at the blastid stage. The second part would be mostly focused on the clinical utility or reporting such findings, especially in the light of the new evidence that we got from the non-selection prospective, the first non-selection prospective study on putative mosaic embryo transfer. So very briefly, I would like to highlight the paramount importance of meiotic aneuploidies in human reproduction and in IVF treatment in general. And we all know well that aneuploidies are for sure the single most important factor that relate with implantation failure or with miscarriages or mental retardation in human pregnancies. And we got recently this very nice evidence from this study that was led by Professor Eva Hoffman, where we got direct observation from a large data set of oocytes donated for research from women across a broad spectrum of reproductive lifespan, showing that aneuploidies can affect between 20, even up to 85% of human eggs, and the rate of aneuploidies, as you can see in this figure, actually overlaps and shapes the U-curve of natural fertility in women between the menarche and the menopause. This is just to highlight how important are meiotically derived aneuploidies for reproduction and how they should be considered as the primary target and of our PGTA strategies. These aneuploidies are for sure coming in a constitutional form into the embryo. That means that all the cells will have this type of abnormality and are not expected to pose any major challenges for interpretation and diagnosis. We got also consistent data from decades of investigation, cytogenetic investigation in human pregnancies, where at the time of amniocentesis we've been always observing that meiotically derived aneuploidies, particularly from the egg, they are for sure the most important and prevalent genetic abnormalities also at this stage, while post-zygotic error leading to mosaicism have been commonly observed at very low frequency in all the cytogenetic studies during the pregnancy. Again, reinforcing the concept that meiotically derived aneuploidies are for sure the primary consideration for our PGTA strategy and development. So let's have a look now more closely to the issue of mosaicism, and I would like to spend a couple of slides to set a clear definition of mosaicism and also to make some clarity about some common myths and misconceptions that are often encountered when dealing with this topic, especially in our field. So mosaicism refers to the presence of genetically distinct cells within an organism that result from post-zygotic mutational events. We may have two different types of mosaicism, depending on the specific genetic defects that is occurring during a mitotic division. We may have the genetic mosaicism that is a variation in the DNA sequence of more CMVs, which is the most common form of mosaicism in the adult stage, and where we have actually no or very little information about the occurrence of this phenomenon in preimplantation embryos. Then we have cytogenetic mosaicism that is involving large-scale chromosomal abnormalities that may be present in a mosaic state, especially at the blastid stage. And this is the most common form of mosaicism that we debate in our field and in the context of PGTA. So here we have the two most important forms of embryonic cytogenetic mosaicism that we should always acknowledge and distinguish when reading the literature and the studies. We can have an aneuploid mosaicism constitution, that means meiotic plus mitotic errors that are present into the same embryos. So in any case, in this configuration, we have that all the cells are abnormal, and this is not a big issue or challenge from a diagnostic perspective, because at the end, all the cells are abnormal and the embryo is fully aneuploid. But the most challenging form of mosaicism is the diploid aneuploid mosaicism, where we have a mixture of both normal and abnormal cells that are present within the same embryo, and depending on where we take the biopsy, we may get, of course, different results. So among the most common myths that we have, or I mean, in my experience, I've been encountering when debating about mosaicism, is that we easily and incorrectly translate some information from different time points of the human life cycles. For instance, while we are accumulating evidence that in the adult stage we are all mosaic, all of us we are mosaic, we have also recent observation from skin cells that 30% of them are abnormal in almost all individuals. We sometimes translate this information to the preimplantation embryos, which is, of course, not really exactly the same stage of life. So we have, for sure, we know that our genome is not static. It does change constantly through development. And we also know that there is a linear association between the number of mitotic events that we have undergone from fertilization onward with the incidence and risk of mosaicism. So according to this, we know that the preimplantation stage is most likely the less subject state for the risk of mosaicism, the period of life where we have the lowest risk for mosaicism. Another common belief that we often encounter in our field is sometimes we hear that our preimplantation embryos undergo frequently in self-correction. That means that these embryos become normal. They start as an abnormal embryos, as aneuploid embryos, and they become normal during the progression through the preimplantation window. So self-correction is an outcome that we can actually measure with good accuracy, because the meaning of self-correction is that we have an aneuploid zygote that, in this case, as you can see in this slide, for instance, let's consider it's a trisomic embryo because it has inherited 2 chromosomes from the egg and 1 from the sperm, and during the self-correction event, it becomes disomic. Disomic during this self-correction means that in a population of cells, of ensuing cells, we should expect to have uniparental disomy by definition. And this type of chromosomal abnormality, UPD, has been measured back in 2014 by the group of Richard Scott and Nathan Treff in a very large data set of human blastocysts, showing that the UPD incidence of the blastocyst stage is around 0.6%, meaning that this embryo self-correction event is not supposed to happen very frequently during the pre-implantation development. And once we have a meiotically derived aneuploidy, this is supposed to be fixed through development. And another common interesting finding that we have from looking at the pregnancies, both spontaneous and IVF-derived pregnancies, is that infertility and IVF procedure do not seem to increase the incidence of chromosomal mosaicism in our pregnancies. We have both data coming from for all chromosome aneuploidies as well as for small CNVs, showing that IVF-derived pregnancies absolutely show the same rate and value of true mosaicism as compared to spontaneous pregnancies, which is around 0.3, 0.4% of our pregnancies, meaning that the frequency is very low. We are not increasing the risk of mosaicism by IVF procedure, and in general, we can thus consider that we are not under an urgent pressure or need to increase and modify our technologies to increase our sensitivity toward mosaicism detection in PGTA cycles. So how is the mosaicism diagnosis obtained during the course of blastocyst stage PGTA treatment cycles? We all started from an assumption. This assumption is that intermediate chromosome copy number values unequivocally predict mosaicism in blastocysts. And in order to develop the methodologies for defining mosaicism in single trophectoren biopsies, we all been based on the analysis of cell line mixture models. So we have been mixing fibroblasts, usually normal and abnormal fibroblasts in different concentration, and setting the algorithm and the thresholds for calling mosaicism then into the clinical setting on trophectoren biopsies. But this, of course, I don't need to tell you that working on cell lines or on diluted genomic DNA it means working in a very stable environment that is completely different from the extreme variability that we may be subject during our clinical work. And all these considerations may clearly explain why we had very inconsistent data reporting over the last few years about the incidence of mosaicism in human blastocysts, ranging from 2% even up to 40%. This is, of course, not a biological difference between the embryo, but all these inconsistent data reporting can be explained by the different methodologies and criteria used for calling mosaicism in human embryos. So, for instance, while we work with in the clinical setting, we may be subject to a lot of variability coming from good quality blastocysts, the use of poor quality blastocysts. We may have trophectoren biopsies with different cellularity ranging from 2 cells up to 15 cells. We may also be using different reagent batches, buffer volumes, or we may also experience variability from shipping condition and also the number of reads that each NGS experiment is assigning to each sample. And at the end of this, there is also some kind of human interpretation variability. For instance, if I highlight this profile plot, I can interpret this profile plot as a no result, while other geneticists may report this as chromosomally mosaic. But I think the largest variability, source of variability, has been the use of different thresholds for defining the mosaicism category in our field. In particular, we should understand that in any case, when running 100, let's say, many experiments of uniformly euploid embryos or uniformly aneuploid embryos, we always have to acknowledge that even in best condition, we should expect to have some kind of variance and variability from the mean value. That means that we should expect to have some euploid embryos that will follow into the mosaicism box just because of physiological experimental variability, as well as we should expect to have some uniformly aneuploid embryos that will be classified as mosaic just because of experimental variability. However, when using very stringent criteria for mosaicism classification, such as between 30 to 70% of variation, this problem is mitigated, is kind of under control, and the typical mosaicism rate that is reported with the use of this threshold is between 4 to 5% of the whole population of human blastocysts. But when we use less stringent criteria, such as defining mosaicism between 20 to 80% of variation, it is true that we are increasing the sensitivity toward mosaicism classification. As you can see, all the yellow dots that are the true mosaic embryos are correctly classified as mosaic, but we lose accuracy and precision in what it is the primary objective of our analysis, that is accurately classifying euploid and uniformly aneuploid embryos. As you can see here, many more by lowering the stringency of these thresholds for mosaicism classification, we are actually mixing up much more embryos, many more embryos into this category of mosaicism. And this is clearly explaining why we have this inconsistent data reporting from the literature, just primarily because of the use of different thresholds for PGTA aneuploidy category definition. Then we have also to acknowledge some more biological source of variability, and that is, for instance, you know that mosaicism can show up at the blastocyst stage in different configurations, such as it can be confined to the SEM, to the trophectoderm, or the abnormal cells can be spread across all over the blastocyst. But pending, and by definition, when we are analyzing an heterogeneous samples, depending on where we take the biopsy, we may have different outcomes. And this is not a kind of technical error or false positive, false negative errors. This is pure biological variability, and that is usually called a sampling bias. And that is also experienced during the typical invasive prenatal diagnosis that we have established from decades in cytogenetic investigation of pregnancies, where when we perform a CBS or an amniocentesis, we always acknowledge this potential biological limitation, that is the sampling bias. A diagnostical error, on the other hand, is reporting mosaicism with exact percentages, and this is something that is very frequent in many practices worldwide. And this is, I mean, conceptually wrong, because we don't know the number of cells that we collect in every trophectoderm biopsy, so we cannot provide exact mosaicism rate. For instance, if we collect 2 cells from an embryo from a trophectoderm biopsy, we can never we can't report 30% of mosaicism rate. Is conceptually wrong. And then we have other biological sources of error when attempting to define mosaicism in TE biopsies. For instance, there may be the profile plots that are associated usually with mosaicism, but may actually also overlap with different genetic configuration of the embryo. For instance, if we have a polyploid embryos with 1 extra missing chromosome, then we should expect to have exactly the same profile plot of true mosaic embryos. And also the opposite way around is true, because even if we do not see mosaicism, this can be equally present within the blastocyst. In this case, for instance, you see a complete normal profile plot because we have been sampling in the same trophectoderm biopsies both trisomic and monosomic cells for the same chromosome, and this will result in a completely normal profile plot. So these are all arguments that should be taken into consideration when developing consent form and genetic reports in PGTA, and should be clearly discussed with patients that even if we do not see mosaicism can be present, and I will come back to this point near to the end of this presentation. So, but even if we are dealing with a very challenging environment, we still have to put our efforts in order to arrive some reliable estimates for mosaicism in blastocyst stage human embryos, because this is really fundamental information for patient counseling, and also very important. It's a very important question, by the way, to answer from a basic research perspective. So in the pre-NGS period, as I call it here, when we were using methodologies and technologies with slightly lower resolution toward chromosome variation, so microarray technologies or qPCR, we had some reports reporting about around 4 to 5% of human blastocysts being chromosomally mosaic, following the common study design of blastocyst disaggregation, where we were disaggregating the blastocysts down to one ICM portion and multiple trofactorial biopsies. But the headache started during the NGS period with NGS transition, where we start experiencing very discordant data about the mosaicism incidence at the blastocyst stage. But all these headache can be resolved by looking at the mosaicism threshold that are applied for instance, for making concordant studies between the trofactor and the ICM. If you go carefully through all the studies, you can see that all the studies that have been using 20-80% thresholds for mosaicism classification, they have been consistently reporting very low poor performance of the trofactor in predicting the ICM chromosomal constitution, while all the studies using non-mosaicism classification, so just reporting meiotic abnormalities, or the ones using less stringent criteria for mosaicism classification, they have been consistently reporting a predictivity of the single trofactor biopsies toward the ICM in the range of 95–98%. So very simple consideration that I can make here is that, and you can, I think, easily agree that is including and incorporating some diagnostic criteria in our practice that is lowering the predictivity of our diagnostic sample, which is a trofactor, versus our target sample, that is the ICM, doesn't make that much sense. This analysis is confined and is restricted just for all chromosome aneuploidies. So here I just want to spend a couple of minutes on our last study on this topic of multifocal biopsies from a single blastocyst. I think is one of the largest investigations reporting data about 319 biopsies, where we've been taken like the ICM plus 4 different trofactorial portions from the blastocyst. And as you can see here, for what concern all chromosome aneuploidies, in 96% of the cases, we observe in a blinded analysis exactly the same chromosomal configuration across the 5 different biopsies collected from the blastocyst. While in just very few instances and chromosomal patterns were consistent with a mitotic or mosaic configuration, around 3 to 4% of them. Separate consideration is required for segmental aneuploidies. With the same study design, we observed that around 70% of segmental aneuploidies are indeed coming with a mitotic or mosaic configuration of the blastocyst stage. And this is actually telling us 2 things. One, that when we make studies in order to investigate the predictivity of trofactor biopsies toward the remaining embryos or the ICM, we should consider that by definition, segmental aneuploidies will be discordant most of the time, because only 30% of them were consistent with a meiotic configuration. And the second interesting point that we can take from this is that when there is a true mosaicism into a blastocyst, true mosaic configuration, we can actually see it by performing a multifocal biopsy study like this, something that we haven't seen for all chromosome aneuploidies. And finally, the last basic research study that I would like to comment with you is this effort by Nina Pavlovich and colleagues that was published last year in Human Reproduction, and where they've been assessing the predictivity of a single trofactorial biopsies versus the embryonic outgrowth on day 12 of development. As you can see here in this summary, you have that euploid and uniformly aneuploid configuration were 100% concordant with the ensuing embryos on day 12 of development, while the mosaicism consistent finding show a positive predictive value of around 6%. And I cannot agree more with the author conclusion, where they suggest that if removing structural and mosaic abnormalities from our PGTA diagnosis, the accuracy increase of around 100% with no false positive and no false negative results. So this takes me to my first summary. So meiotic aneuploidies are for sure the main abnormalities in human embryos, while true mosaicism is a very relevant in both spontaneous IVF pregnancies and reliable estimates for mosaicism in human blastocyst stage. The human blastocyst stage is around 2 to 4%. Mosaicism can be easily overestimated by using inaccurate methods and weak criteria for classification, and we have many several technical and biological consideration to make that will show and will overlap and cannot be distinguished by current technologies and will be overlapping with a mosaic profile. That's why, in case we report mosaicism in PGTA cycles, it is absolutely necessary to report it as putative mosaic, where putative can be, is an adjective to mean that it is generally thought to be or to exist, even if this may not really be true. And to a more wider extent, I think a better summary of this part of the presentation is that by trying to increase our sensitivity toward mosaic abnormalities, we are really taking the risk of losing the focus from the big picture, from our primary analysis that should be looking at uniformly aneuploidies with the highest accuracy that we can get. So in the second part of my presentation now, we can go and have a look what is, or if there is any clinical utility of reporting these mosaicism consistent finding in our practice in PGTA cycles. And I think that it is worth to start this discussion by mentioning that in case we report mosaicism and when we report mosaicism in the clinical setting during the course of a PGTA treatment cycles, this comes with some really important consequences for the patients and for the general setting of the IVF treatment. So first of all, if we report mosaicism, we need to anticipate that the couple is willing to go and will go through an independent session of genetic counseling, and we'll have to evaluate the embryo transfer in a context of higher anxiety, distress, higher cost, and potential wastage of precious time. Reporting mosaicism comes also with a consequence of increased adoption of amniocentesis that is today recommended in order to confirm the mosaic finding in the subsequent stage of pregnancies. And I think the most concerning point is the potential wastage of embryos that we may have by reporting mosaicism, especially with less stringency, because these diagnoses can be perceived by patients as that their embryos are genetically abnormal, and they may be deciding to not to transfer, not to use these embryos for the IVF treatment. So if we look, for instance, at the largest study reported by Monet and colleagues in 2017, we can see that less than 3% of the whole 4,900 embryos that were classified as mosaic have been transferred back to the patient. That is clearly highlighting a very concerning and alarming potential wastage of embryos. So let's have a look if this is justified by evidence. So here in this slide, you have a table that is summarizing all the studies, the retrospective studies that have been reporting clinical outcomes following the transfer of putative mosaic embryos. All these data have been delivered and produced by using less stringent criteria for mosaicism classification between 20 and 80% of variation. And as you can see here in this picture, in these bars, you can see that mosaic embryos associated with a slightly lower live death rate and a moderate increase in the early abortion rate. There are some alternative explanations to explain why putative mosaic embryos have lower performance compared to, let's call them, purely euploid embryos. For instance, one of these is that it is still a possibility that some of these putative mosaic embryos were actually uniformly aneuploid embryos that were classified as mosaic just because of a normal variability of the aneuploid signal that went through the mosaic category. And my colleague Laura will have a presentation at this year upcoming ASH annual meeting about that. And I can anticipate that 36% of uniform aneuploid embryos have one TE biopsy in the range of 58%. So this is a possibility to consider. And of course, especially for the high-level mosaic, we should really pay attention to transfer these embryos without any additional confirmation about the fact that these embryos are not uniformly aneuploid. And the second alternative explanation for the lower performance of putative mosaic embryos is the selection bias of the patients where we've been observing and recording the outcomes of these mosaic embryo transfer cycles. Because, as you know, according to the current workflow in PGTA, when reporting mosaicism, the first option is always to transfer euploid embryo. And by so doing, we transfer as first option euploid embryos. The good prognosis patients get pregnant with these embryos. And actually, the patient population where we observe the outcomes of a mosaic embryo transfer is a very highly selected patient population of poor prognosis that have already failed with several euploid embryo transfer cycles. So the poor performance of these embryos can relate more to the patient factor rather than to the genetic signal. So in order to check these hypotheses and to exclude any potential selection bias in our analysis, we've been performing back in 2017 this 3 years prospective non-selection study, where the putative mosaic embryos up to 50% of variation have been reported back to our clinics and patients as normal, at the same strength as we were reporting a purely euploid embryo. And by so doing, we've been able to remove any bias related to the selection of patients, and also we have been able to get a definitive assessment about the reproductive performance of putative mosaic embryos in the range of, up to 50% of variation. So for the purpose of this study, we've been using the ion requisite PGTA workflow with some minor modification from our PGTA team. This methodology has been validated also for detecting mosaicism on cell lines and diluted genomic DNA, and work equally as for any other NGS-based technologies for detecting mosaicism in this environment, in this more experimental environment. And here you have the demographic and cycle data of the enrolled patients over the last 3 years that have been recruited in 5 different clinics in Italy that I really would like to acknowledge and thank for their efforts and for trusting in our concept that we would need first and primary class 1 data for addressing and investigating the clinical performance of putative mosaic embryos before calling them as mosaic. As you can see here, we have investigated 845 PGTA cycles where at least one euploid or putative mosaic embryos was recruited and was obtained during the PGTA cycle. This is a typical population of advanced maternal age, as you can see, 39.5 mean female age. And the overall aneuploidy rate was 41%, and the euploidy rate was 58% in this population of advanced maternal age patients. Here you have a breakdown of the euploid embryos, the ones that were originally defined as euploid. I just want to point out here that this is nothing to deal with the policy that in Igenomix we are using for reporting mosaicism. This is just a post-transfer analysis of the raw data, the raw signal that we got from the NGS profiles, where we could divide the euploid embryos in the purely euploid without any variation up to 20%, in the putative mosaic category, low mosaic category between 20–30%, or in the moderate mosaic category with a variation, chromosome variation between 30–50%. And here in this picture, you can see all the data points across all the chromosomes that we obtained from these 2,784 two-factor biopsies. And here you have the main clinical transfer outcomes. As you can see, positive pregnancy test, biochemical pregnancy loss, miscarriage rate, live birth rate were all similar. We didn't show any difference between the 3 PGTA euploid categories. We have checked also these analyses in a multivariate logistic regression model where we could check and control for confoundings. And also the main neonatal outcomes that we could recall, such as the birth weight, the gestational period, were all similar between the three study groups. We also tried to develop some additional metrics for checking whether these mosaicism consistent findings were associated with any of the main clinical outcomes. So as you can see in this table, the mean mosaicism rate, mean number of mosaic chromosomes, the incidence of complex mosaic were all similarly distributed in these three principal outcomes, such as the biochemical pregnancy loss, miscarriage, and the live birth. And I think one of the most interesting observation that I wanted to highlight here is the potential implication that we may have on cumulative live birth rate if embryos or putative mosaic embryos are indeed perceived as genetically abnormal and not considered for transfer by our patients and clinicians. In particular, when looking at the data from mosaicism rate reporting above 20% of variation, you should expect to have a reduction in the cumulative live birth rate, because if these embryos are not utilized of around 36%, which is something that is worth considering when designing our PGTA strategy. So here we have some limitation, of course, as for all studies. First of all, the study was powered to detect meaningful differences of live birth rate of around 5%, but looking at the actual data, the study is powered to detect even lower differences than this, to exclude lower differences than this. And probably the main limitation of our study that we could not perform amniocentesis in a significant proportion of the ensuing pregnancies for both euploid and putative mosaic-derived pregnancies. And I can explain you the reason why is that we couldn't give a strict indication for amniocentesis that, as you know, is an invasive procedure and is expected to result in 1 out of 200 cases in a iatrogenic abortion, while at that time, and still today, the positive predicting value for mosaicism diagnosis performed in a single trophectomy biopsies, that means how many of them have been confirmed in later stages of pregnancies or in the newborns, is just accounting for one single case report that was recently published by Professor Karaman from Memorial Hospital of Istanbul. So all in all, we had so far thousands of putative mosaic embryo transfer, and only one consistent finding of mosaicism was detected in the newborn. So at that time, we couldn't give a specific indication for mosaicism to our patients getting pregnant with these analysis, because the miscarriage and abortion risk was higher than what we can expect to be the mosaicism risk in a pregnancy. And also, the negative predicting value is another important parameter and diagnostic measure to consider. So in this case, the negative predicting value relate to the extent on how we can decrease the incidence of mosaic pregnancies following the transfer of purely euploid non-mosaic embryos. And at present, we don't have any of such information and data from the literature. And considering the general incidence of mosaicism in human pregnancies, to get robust evidence on this negative predicting value will take very large sample size. We have just a few evidence of the opposite, actually, that came recently from this publication, where you can see that the use of high-resolution NGS and calling mosaicism, even with less stringent parameter, between 20 to 80% of variation, actually resulted in 60%, around 60% of the ensuing product of conception being mosaic. So, as I was anticipating you at the beginning of this presentation, it is clear that even if we avoid the transfer of putative mosaic embryos, we cannot exclude the occurrence of mosaicism in the resulting pregnancies. And the final comment that I would like to make on this subject is related with the decision of and the prioritization strategies of choosing always the transfer of euploid embryos as a first option. And this was also endorsed and supported by 2 different scientific societies. But even if there is any evidence that this is beneficial for the patients, we may have even situations where following this guideline can be even harmful for the patients, particularly in this case where you have that the euploid embryos is the poor morphology blastocyst, while the good quality embryo is has had a diagnosis of putative mosaicism, and we transfer first the poor quality embryos. This is absolutely, I don't think that this can be considered as a good practice, good medical practice, because we know in several studies that the very poor quality blastocysts are associated with a higher miscarriage rate, which is independent from chromosome abnormalities. And also in terms of time to pregnancy, this is probably not also the best option for the patients. And probably this change in the embryo prioritization can be one among the reasons that explain why the STAR trial has failed to show benefit of PGTA, while we had decades of investigation, many different RCTs, case control studies, even some of them very large, all consistently showing that PGTA is effective and it's very efficient way in order to improve live birth rate per embryo transfer, reduce miscarriage risk compared to untested blastocysts. So I think that using these less stringent criteria for mosaicism classification not only lower the predictivity from a single trophectoral biopsy toward the ICM, but yet also not clinically validated at present. And this is my very last slide, my conclusion and take-home messages. I can conclude that PGTA is for sure a very effective technology for detecting meiotically derived aneuploidies and improving IVF outcomes. Of course, it has some limitation, as any testing procedure, as any genetic testing technology, such as the biological limitation of mosaicism, that as we do for amniocentesis or CVF, should just be acknowledged in consent form and genetic reports with all the consideration that I made with you in this presentation. And then a more general consideration and conclusion is that we have also and always in our practice, when we incorporate new criteria, trying to define and deliver accurate positive negative predicting values before incorporating new diagnostic schemes or criteria in our strategy, in our diagnostic strategy. And these predicting values should not be looking at the predictivity into the single trophectoral biopsy, because our patients, and I think also us, we are not interested in predicting the genetic constitution of a trophectoral biopsy, but we are interested in predicting the chromosomal constitution of the resulting pregnancies, or to more widely extent, the chromosomal health of the ensuing babies. The clinical utility of reporting mosaicism in this context, especially with low stringency for classification, I think is definitely yet to be proven, while some counterproductive effects can be certain. And also, when we hear that this finding has to be reported because they are seen into the NGS profile, this is absolutely not recommended by any validated framework for reporting genetic findings, where we know that, and there is wide consensus in genetics today, that we have to report genetic findings not just because we see them, but because they are associated with clinical benefit and utility for the patients. And this is my last comment and point. So I want to thank all Igenomix, in particular Carlos, for supporting this study from the beginning, all the team of Igenomix Italy. They are wonderful and very passionate in their work, as well as a particular thanks goes to the clinics that for this specific study, they really believe in it, and they also put all their efforts for making it happen. And thank you, of course, for your attention. I'll be happy and honored also to take your question and discuss further this topic with you.
Thank you for your wonderful presentation, full of data, full of knowledge. You made a great background for us to understand your study. So thank you also for your first part of your presentation. We have here some questions regarding what you consider low mosaicism. Why do you consider that the result of mosaicism cannot be right if it's always an artifact caused by the genetic technique or by the biopsy? So I think you told a little bit about it, but if you want just resume, low mosaic and technical artifact.
Well, it's very difficult then to say what is the main reason that causes a low mosaicism consistent profile. So, but the first question we want to answer in this study is whether this relates with any outcomes and performance, reproductive performance of the embryo that we have in our hands. And as I think I show in this study, we show in these studies there are not major consequences of this profile that are consistent with low mosaic for what concern reproductive outcomes. So what is the main reason behind that? I don't know. I'm sure, and I can tell you that the lower is the stringency of the criteria that you use, and I think I stressed this concept as well. The higher is the impact that you have in the PGTA aneuploidy categorization. In particular, the lower is the stringency, the higher is the risk of reading mixing up many different aneuploidies type into the same category, such as the uniformly aneuploid and the mosaic, the true mosaic. So I think a reasonable way in this context is to minimize the issue of technical variability that we may have in our daily life, is to really narrow the range and indeed this translates in very in a lower rate of mosaicism reported in general, and also these embryos at the end, the low mosaic implant and deliver at the same rate as purely euploid embryos.
Okay, great. Thank you. So now I'm going more related what regards your last study that you present. So we have here a question saying, with your study, did you find any difference in implantation outcomes between mosaic and euploid embryo? This is the first part of this. So it's implantation outcomes, euploid and mosaic. And the second part, can you comment on the karyotype abnormalities and UPG occurrence in the fetus in neonates with the mosaic embryos transfer?
Yeah, very good questions. For the first, we didn't observe any difference in implantation, miscarriage, biochemical pregnancy loss. They really behave exactly the same as purely euploid embryos without any variability, experimental variability. For what concerns the second question, I think this is a very good point that we are trying to answer now. We are in the way of performing karyotype analysis from the babies that we obtain during the course of these studies, and so I think soon we will be able also to provide some more information about the potential presence of still some persisting chromosomal defect in the three categories or the two categories, the fully euploid or the fully aneuploid embryos. So this is something that is at the moment is ongoing.
Yeah, the UPG is something that we have to wait, right? So this you cannot measure right now.
No, we cannot measure right now, but as I explained before, yes, during the presentation, we couldn't, unfortunately, we couldn't give a very strict indication for amniocentesis in this study.
Yeah. Thank you, Antonio. I'm going on, okay? I'm shooting you because we have a lot of questions here. So can you comment the utility of using SNP array data to distinguish mitotic and meiotic aneuploidy in the context to improve accuracy of predicting mosaicism?
Yeah, this is another good question. Yes, probably one possibility, yes, can be to use NIP array on professional biopsies in order to detect just uniformly derived aneuploidies. But I don't think that there is that much need. I think we can still working in a very effective environment for distinguishing and detecting primarily uniformly aneuploidies with NGS that on the other side has many more advantages, including the throughput capabilities and possibility to keep significantly low the cost and increase the access to this practice for many more patients.
I have one question. This is mine. Once we know that a mosaic, it's mitotic or meiotic, it could be different. Of course, if it's meiotic, the chance to be the full abnormality, it's higher. I know that. But once transferring a mosaic embryo, is there you imagine any different outcome from the mitotic mosaicism? So if it's true, if persists, even being a mitotic mosaicism, it persists, could be a different outcome to the baby.
So can you formulate again? So in case we have a mitotic.
Because the question using the SNP array to distinguish mitotic and meiotic aneuploidy, we know that it's meiotic aneuploidy, it should be full aneuploidy. The chance to be 100% cells aneuploidy is higher. Yeah. But we don't avoid the possibility to have a mosaic if this embryo receive an abnormality from the meiosis.
I've got your point, and yes, for sure, in case we had a very early event of mitotic errors, and the abnormal cells will be the most prevalent across the whole blastocyst, and for sure they will have a clinical meaning. Of course, there is the risk of missing this type of abnormality by using SNP array, while with NGS we will still be able to identify them.
So in the end, we give an information that maybe it's not related to the outcome. Anyway, again, more and more questions regarding the follow-up with the babies. So I think you answered already that you want to do the karyotype for that. We have a lot of comments saying, excellent presentation. Thank you so much for your data. I have another here saying, asking regarding the genetic counseling. So should the genetic counseling explain mosaicism when you say that you can transfer low mosaics the same than euploid? Should be done during a pre-PGT, or what do you think about the genetic counseling should cover with the patients regarding this subject?
Well, I really hope, and this is our hope, that this data here that we deliver will increase a little bit the confidence also both the genetic counselors, the physicians, and especially the patients to consider the transfer of these embryos, at least from what concerns the reproductive outcomes with the use of these embryos. Then, of course, in time, we will need more information about the karyotype of the baby, some probably long-term follow-up in order to gain more conclusive evidence. But at least the hope here is that we will have more embryos with a low mosaic profile or putative mosaic profile that will be considered more for clinical use. Yes, of course. I think that genetic counseling in this process is becoming really the fundamental part of the PGTA, because it should be made clear that in any case, mosaicism, even if we do not transfer putative mosaic embryos, mosaicism can be present in the pregnancies, because we may fail to detect it in the TE biopsies, but it may also occur after the time of implantation, so in later stages. So in any case, mosaicism can be still an issue, even if decided not to transfer such embryos. And and in general, we've been always transferring embryos in IVF, and we never had, or euploid embryos with RACGH, with qPCR, and we never had such high rate of mosaic pregnancy. So in general, I think we should really mention and this issue in our field because I mean it's just a matter. I think all the issue comes just because of use of really not correct criteria for classification that have just led to overcoding and overestimating this problem in our field.
Yeah, okay. So as our timing, it's almost done. I have just my last question here for you. I will read it. So you say that the mosaicism risk in pregnancy is 1 in 1,000. So that's the reason you make a comparison with the prenatal test in your slide. So, and you consider the mosaic embryo in a pregnancy, mosaic embryo reported in a pregnancy. So the questions are, how many mosaic babies were not reported in studies? And would you consider mosaic POC also in this calculation?
Well, what was the data that I delivered, around 1,000, refers to the number of mosaic findings that were detected in the PGT analysis and were then confirmed in later stages, like during the pregnancy or at the time of birth. And there is only one case report so far, but we know that thousands of embryos with a mosaicism-consistent diagnosis have been transferred. This was the positive predicting value of the diagnosis that have been associated with a trophectoderm-based mosaicism analysis. So in general,
I think that's the reason they asked how many was not reported. So this is the first.
Yeah, for sure. Probably there will be some that have not been reported. We know that the incidence of mosaicism, true mosaicism, the one confirming pregnancies, is around 0.3%. So yeah, there's still a possibility that we had, but we don't know if the mosaic cases that we lost probably in the first moments of the pregnancies are coming from the purely euploid embryos or the putative mosaic embryos. So we are trying to address now this issue in the stage where we can, that is in the newborns. And that is at the end what matters. At the end, as I trying to explain in my last slide, I think all our diagnostic performance measures should be based on the chromosomal health of primarily on the ensuing baby.
Great, Antonio. I really want to thank you for being here with us today. You have more than 450 people following you, the whole presentation and the questions. We have much more questions that I couldn't make for you. I will address to you the questions, and maybe you can discuss this. Okay.
Yeah, my pleasure, and thanks for following the webinar. Really appreciate that.
Okay, thank you so much. Bye-bye, everyone. Thank you. We see on next Thursday again.